morning, everyone. Over the past 10 years at NIH, it's been my privilege to work with outstanding colleagues and an array of remarkably talented scientists like those from whom you're about to hear on strengthening support for basic behavioral and social sciences at NIH, and also on leveraging those basic science insights and advances to support translational efforts to promote health and prevent disease. This panel is around that theme, which is really emphasizing that first pillar of the new OBSSR strategic plan, strengthening the synergy between basic and applied science. One flagship initiative, the NIH Science of Behavior Change program, is an exemplar of this kind of integrative work. That's a program that seeks to leverage emerging mechanistic insights from transdisciplinary domains of behavioral and social science, like social and affective neuroscience, behavior genetics, behavioral and neuroeconomics, and behavioral medicine to search for and validate intervention targets for behavior change. It engages scientists who can identify the social, interpersonal, psychological, and biological mechanisms that drive behavior. It engages individuals who work in the lab and in the field to elucidate the conditions under which these mechanisms operate. And it engages scientists who can apply these insights to develop more potent and implementable behavioral interventions. The fundamental philosophy that drives the Science of Behavior Change program is also at the heart of the work that's being conducted by the individuals in this morning's panel. It's work that seeks to overcome that artificial separation between basic science, where mechanistic intervention targets can be identified, and clinical or interventional science, which seeks to modify those targets to promote health, promote optimal human development, and slow disease progression. So it's my distinct pleasure this morning to present the speakers in today's panel, individuals whose work exemplifies this synergy between basic and applied behavioral and social science, aimed at elucidating the pathways linking social, behavioral, and psychological factors to health across the full life course. The goal of this research panel is to showcase exciting lines of research illustrating the complexities involved in identifying potentially modifiable behavioral or social mechanisms on the causal pathway to health and disease. These presentations will reveal how observational and interventional studies can serve to advance our understanding of how social and behavioral factors get under the skin to impact health, and how behavioral processes can serve as promising intervention targets. So we'll have 3 speakers into this morning's panel. Each of the speakers will have 20 minutes to speak, and then after all the talks have been completed, we'll have a 15-minute Q&A with the audience. So I'll introduce each speaker in turn, and I think perhaps you all want to stay down there where you can best see the talks and then come up for your individual presentations. Our first speaker is Dr. Jean Brody. Dr. Brody is Regents Professor and Director of the Center for Family Research at the University of Georgia. He's a developmental psychologist and a prevention scientist interested in the social and biological determinants of health and well-being among low SES youth. He's an internationally recognized expert on the risk and protective mechanisms that forecast substance abuse and other problem behaviors among rural African American youth. He has translated his findings into efficacious preventive interventions targeting family and community factors such as parenting styles and extrafamilial support systems for youth ranging in age from age 11 to 19. Dr. Brody is the author of over more than 350 publications, many of which focus on African American youth development or substance use prevention. His work has been supported by NIDA, NIAAA, and NICHD, and his recent work on a phenomenon he and his colleagues are calling skin-deep resilience challenges us to think more carefully about how to foster both positive coping and robust health in individuals from disadvantaged backgrounds. So it's my pleasure to introduce Dr. Brody, whose talk will be a glimpse of the research sponsored by the Center for Translational and Prevention Science.
Great. Good morning. So today, my talk is actually going to be a demonstration project. And the project is designed to help you get a glimpse into the benefits of embedding neurocognitive, inflammatory, immune, and measures in the brain into the kind of work that we're all interested in: longitudinal epidemiologic designs and in prevention designs. And so my goal is to demonstrate this with some real live data so you can get an idea concretely the kinds of questions we can answer by doing this work. This work, however, requires not just individual groups of investigators, but transdisciplinary groups from multiple backgrounds who come together to ask questions that any individual investigator themselves would not be able to answer. So today, what I want to do first is, in this demonstration project, tell you who we're talking about. Our data that you're going to see that exemplifies what I want to demonstrate for you is drawn from people who are representative of the Black Belt that stretches from South Carolina, Georgia, Alabama, Mississippi, and Louisiana. The 623 counties in this area are some of the poorest parts of our country. 35% of our nation's poor live there, and over half of the children who grow up there grow up under conditions of economic hardship. The actual data in my presentation today comes from 38 counties in Georgia who mirror pretty closely the demographics of the Black Belt that I just described. Most of the caregivers who participated in the research I'm going to talk about today work either one or two jobs at very low wages. They are best characterized as working poor, and the vast majority of the caregivers are highly competent parents who we refer to as heroes. So I want to give you some examples of the kinds of questions you can answer by embedding different kinds of data into a longitudinal prevention trial. In our first exemplar, we took a sample of 500 African American youth who began our study at age 10, who are now age 25, and were tracked as part of that longitudinal analysis through the Great Recession that started in 2007 and officially ended in 2010. And we obtained a variety of markers on each of these 500 youth. At age 19, we obtained overnight urine voids from which we got the stress hormones cortisol, epinephrine, norepinephrine. We drew blood to get C-reactive protein, a marker of systemic inflammation. From that blood, we also got measures of accelerated epigenetic aging. We took their blood pressure and their BMIs. And so the question we wanted to ask here, and as an exemplar of kind of some insights you're able to discern from looking at a natural experiment like this, is at 2007 and 2009, some of the youth didn't experience any economic downturn, experienced no economic hardship. Others lived in economic hardship across the Great Recession, while a third group evinced a decline in their economic well-being. And what we were able to discern by embedding these measures into that longitudinal data collection was that youth were at higher cardiometabolic risk, meaning they had higher levels of circulating stress hormones, they had higher blood pressure, they had higher BMI, and higher levels of inflammation in their bodies when they lived across that Great Recession at the highest levels of economic hardship. Of particular interest, though, and what was fascinating to us, is that youth who had downward economic mobility during that time evinced higher levels of cardiometabolic risk and accelerated biological aging compared to children or adolescents who did not have any economic hardship across that period of time. But when we asked them about their health, those youth who experienced an economic downturn did not self-report any health problems, even though their biology said otherwise. There is some lore in the health disparities literature that many youth grow up and their communities change and they become enveloped in poverty. And we wanted to test this idea, and we also wanted to test ideas that were drawn from animal models, which said that supportive and sensitive caregiving could protect youth in their neuroendocrine systems, in their immune systems, and in their brains if they were the recipients of supportive caregiving. So on 493 kids at age 11, we took census data, we looked at their neighborhood levels of poverty, and we looked at their same neighborhood levels of poverty where they lived when they were 19. And there were some youth who over that time really did live in environments that were enveloped by poverty, and could that be ascertained in their biology? So again, we looked at their cardiometabolic risk with those same measures of circulating stress hormones, blood pressure, BMI, inflammatory response, and we found indeed those youth growing up in neighborhoods that became enveloped in poverty, below their skin, their biology distinguished them, particularly if they did not have the receipt of supportive caregiving. We could not have ascertained these kinds of questions without the embedding of these markers in our longitudinal sample. In the health disparities literature, there is a hypothesis that exposure to racial discrimination chronically over time reduces life expectancy and because of accelerations in biological aging that accompanies the coping that goes along with exposure to those chronic stressors. And in this study, we wanted to test this hypothesis. So using our measure of accelerated biological aging, indexed via epigenetic aging, we wanted to see across the ages of 16 and 18 in one sample if we could identify a group of adolescents who were exposed to high levels of racial discrimination, and if 3 years later that was associated with accelerated biological aging. And if it was, could we also ascertain whether supportive caregiving could ameliorate those biological changes which actually speeds up aging? And what we found in this first sample is indeed youth who grew up exposed to high levels of discrimination were evinced accelerated biological aging, particularly if they did not have receipt of supportive caregiving. We examined the same hypothesis in another independent sample where from ages 17 to 19 in 223 youth from rural Georgia, the youth reported on their exposure to racial discrimination, and once again, except this time at age 22, we drew blood, we looked at their biological aging for youth who had high levels of parental support and supportive sensitive caregiving versus those who did not, and replicated almost precisely our findings in our first study. So in summary, by embedding these measures of neuroendocrine processes, inflammatory processes, epigenetic processes, along with measures of blood pressure and BMI, we were able to ascertain that indeed there was a signal that indicated that exposure to economic hardship was associated with different kinds of biological profiles under the skin in youth, that changes in neighborhoods also was associated with different kinds of biological configurations in youth, and finally, exposure to chronic levels of racial discrimination was associated with accelerated biological aging. So I'm going to skip over some other data to get to some prevention data, because I think that it's very important for us to show the benefits in our demonstration today of embedding biomarkers into efficacious prevention programs that were not designed themselves to impact health outcomes, but may have health benefits because they enhance supportive caregiving, which our data show can ameliorate stress from getting under the skin. So in our center, we've developed 3 prevention programs. They are developmentally appropriate prevention programs designed for rural African American families. The first one, SAFE, was designed for pre-adolescents. The second one, the Strong African American Teens program, was designed for adolescents. And Adults in the Making was designed for youth and delivered to youth during their last year of high school before they go out into a very challenging and often unforgiving world. Each of these prevention programs were tested in randomized trials with Ns of at least 500. We are still following some of these samples today. And in 2 of the programs, we embedded biomarkers in SAFE and in AIM. And I should say that each of these programs evince their prevention goals for at least 2 years beyond participation in the prevention experience. We've tracked the kids in SAFE and find prevention effects 8 years after participation in the prevention program. In this first paper, in this first demonstration, what I want to highlight here is we drew blood from the SAFE participants when they were 19 years old, and they participated in prevention programming at age 11. And SAFE was designed to enhance supportive caregiving and emotional support as one of many markers. And we wanted to see if if participation in SAFE at age 11 carry forward to impact on markers of inflammatory response, which is particularly important to this population because they have prognostic significance for the development of cardiovascular disease, diabetes, stroke, and hypertension. Secondarily, we wanted to see if changes in supportive parenting during the SAFE prevention trial were responsible for any obtained effects. And what you can see is that those youth who participated in SAFE at age 11, at age 19, they evinced lower levels of cytokines that orchestrate inflammation, and that these effects, not shown here, were mediated by increases in supportive parenting and decreases in harsh parenting. An example of mixing prevention with psychosocial targets and biological outcomes that are relevant for health. The second study addresses an important clinical question for the population of caregivers in rural Georgia. Many of these caregivers are coping with multiple demands and chronic economic hardship, and it kind of rips at their psychological fabric and induces levels of depression. And we wanted to ask in this particular study whether growing up with a depressed parent forecast accelerated biological aging in the youth, and whether participation in SAFE could ameliorate the link between caregiver depression and accelerated biological aging, and if the mechanism for this decrease were reductions in harsh parenting. So what this slide shows is that those youth who grew up with a depressed parent and were randomly assigned to the control condition, those youth at age 20 showed accelerated biological aging, and that those youth in that particular condition, where moms were depressed and they were in the control condition, showed low decreases in harsh parenting, and a subsequent analysis showed that the reduction in accelerated biological aging for those youth in the control condition who grew up with a depressed parent was SAFE's change or reduction on decreases in harsh parenting. I misspoke. Those youth in the SAFE condition with a depressed caregiver evinced larger reductions in harsh parenting, and those reductions in harshness were responsible for SAFE's effects on accelerated biological aging. Again, another demonstration of mixing prevention programming with psychosocial mental health outcomes and a biological endpoint, and accelerated biological aging forecasts early mortality. Finally, we had a study that just came out this past week, which is another demonstration, except this time we focused on neuroscience outcomes in the brain. There's a literature that links brain development to stress, particularly in two areas, the hippocampus and the amygdala. And we focused on two areas of the hippocampus which have in translational studies shown to be particularly influenced by adversity and stress. There's some studies with kids which are pretty consistent with those results. And we wanted to take a look at if kids lived in poverty across adolescence, could that be evinced in their brain volumes in these hippocampal areas of the brain and in the amygdala when they were 25. So in this analysis, we put 115 young adults who participated in SAFE, half who participated in SAFE, half who participated in the control condition, into the scanner, looked at the volumes of these two hippocampal areas and the amygdala to see if participation in SAFE 14 years earlier was associated with larger brain volumes. And as the data show, youth who spent more years in poverty and were in the control condition evinced smaller hippocampal and amygdala areas. Again, we're trying to show here that there is terrific benefit and currency for those of us who are social and behavioral scientists to be able to demonstrate that the processes that we are so able to assess and analyze have implications for markers of health that may carry forward across the lifespan. Thank you.
See a nice picture of his group of co-investigators there. Our next speaker is Pete Gianaros.